Welcome, everyone. Thank you very much for joining us. My name is Yixian Zheng. I'm director of Carnegie's Embryology department. I want to begin today's fascinating program with Dr. Mark Roth by briefly introducing our department's history. The department is one of 6 research departments within the Carnegie Institution for Science. Our department has been at the forefront of scientific discoveries and innovations since 1913, starting with the research on human embryonic development. The current staging of human embryo development is actually based on the department's research. If you just search for a term, Carnegie stage, on Google, you'll find the descriptions of early human embryo development and pictures. The modern biology era of our department was ushered in by Dr. Don Brown, our former director, who succeeded in isolating genes and studying them in the test tube, which enabled DNA cloning. Another of my predecessor, Dr. Alan Spratling, is a pioneer in developing the fruit fly as a stand-in for understanding the shared biology among animals, including us humans. The tools Alan and his colleagues developed have enabled many critical discoveries made in fruit flies, which resulted in 6 Nobel Prizes, including one on the discovery of a branch of animal immune system called the innate immunity. The innate immunity branch is the first line of defense against microbes, including the SARS-CoV-2 virus that causes COVID-19 that affected all of us in the past year. During Alan's leadership, our department enjoyed continued success, including the discovery of RNAi by Dr. Andy Fire, which was recognized with the Nobel Prize in 2006. Our research style is predicated on the belief that original discoveries require exceptional individuals and the freedom to explore new frontiers. Soon you'll see why today's speaker, Dr. Mark Roth, embodies the creative spirit of a Carnegie scientist. As a postdoctoral fellow in our department from 1984 to 1988, Mark worked under the guidance of my colleague, Dr. Joseph Gall. Joe is a luminary in biological research who made great contributions to our understanding of many aspects of the cell. For example, Joe pioneered the study of chromosomes and many other structures in the cell's nucleus. Joe is often regarded as the founder of modern cell biology and was recognized by many prizes, including the prestigious Albert Lasker Special Achievement Award in 2006. Joe will introduce tonight's featured speaker, Dr. Mark Roth. Joe, you can take away now.
Of the many graduate students and postdocs who trained in my lab here at Carnegie, I rate Mark Roth very near the top in scientific creativity. Mark is quick to see the implications of his own work and that of others, and he automatically searches for ways to apply or extend whatever new knowledge he acquires. He frequently leaves others wondering how he got from point A to B in his reasoning, but more often than not, his intuition proves remarkably correct. Mark has made major contributions to several seemingly disparate fields: SR proteins and splicing, autoantibodies, structure of the eukaryotic nucleus, and most recently, suspended animation. The unifying thread among these topics is the search for basic molecular mechanisms to explain cellular structure and function. Mark has tremendous motivation and enthusiasm for his work, which you will soon see. For instance, in his work on suspended animation, he decided that it would be useful to test the effects of carbon monoxide, an extremely dangerous gas. Many people would probably conclude that the hassle in setting up proper facilities outweighed the scientific benefits. But Mark went ahead and as a result uncovered completely unanticipated facts about how the cell deals with anoxia. In short, Mark thrives on uncertainty, novelty, and risk-taking, all combined with hard work and perseverance in the face of challenging odds. A measure of Mark's ability to move into new areas is the number of organisms with which he has worked over the years, in each case chosen for some specific experimental advantage. He has done significant work with amphibian oocytes, cultured cells, C. elegans, Drosophila, mice, and zebrafish. This reflects his ability to focus on the scientific problem at hand and not worry about his familiarity with a particular organism. Mark has shown a remarkable ability to take his findings beyond the narrow setting of his own laboratory. For example, his work on SR proteins led him to develop a screening test for autoantibodies that is useful for detecting autoimmune disease. His work on lack of oxygen and suspended animation, which he will speak about, has profound medical implications. It is even possible that he could devise methods to store living organs for long time periods, thus overcoming one of the major problems in organ transplantation. Mark additionally worked with a group of engineers to develop new forms of microscopy based on Raman light scattering. What I have been describing sounds as if it would require a huge laboratory with a large scientific staff and multitudes of technicians. Quite the contrary. Mark has a very small group and he carries out many of the experiments himself or directly supervises the work of a graduate student or postdoctoral fellow. He is definitely a hands-on scientist who is intimately aware of exactly what everyone in his close-knit group is doing on a daily basis. It is precisely this direct involvement that is the secret of his productivity and ability to remain focused on basic scientific issues. In conclusion, I can think of few others whom I would so readily categorize as both a truly visionary thinker and a superb experimentalist who clearly distinguishes between what can be accomplished and what is mere speculation. Mark is truly an inspiration for us all. Now I will turn the microphone over to Mark himself.
Thank you, Joe. That was a very kind introduction. And I really greatly appreciate all of the help you've given me over the years to do the work that I've been able to do. You and David Prescott, my graduate advisor, truly inspirational for me in my own scientific career. So thank you very much. As the title reads, Adventures in Suspended Animation, I am going to do as Joe suggested. I'm going to take you on a trip, tell you a story about the work that I've been doing in suspended animation. And lest I forget, I'm going to begin by thanking the people who did the work. Some of the current and unpublished work I'm going to mention was done by Mike Morrison, Akiko Owada, and Mary Wick, who are members of my lab now, along with several collaborators and their teams of workers at Harborview Medical Center, Dr. Ron Mayer, Dr. Dan Henning, at UCSF Medical Center, Dr. Jeff Manley, and at University of Alaska at Fairbanks, Dr. Kelly Drew. There's also many, many people who have been involved in the clinical studies that I will mention. And these people are and were and still are employees of Icaria and Faraday Pharmaceuticals. So we're going to cover a large distance intellectually. For those of you who are scientists and want to hear about the mechanism of action and the chemistry and the details, you won't hear it today. Please feel free to ask questions. I'll try to answer them at the end or whenever you write them, because we're going to take a kind of a tour through the efforts that I've been making over the last many years to think about suspended animation and trying to understand it fundamentally in a way that can allow for the ability to possibly change critical care medicine. That's the goal. So here we go. So I first became interested in the idea of studying suspended animation because, like many of you, I read in the newspaper stories of miraculous survival stories. So for example, here's one that I read in 2015. A disgruntled 15-year-old got into the wheel well of a jet in San Jose, California, and flew to Hawaii for about 5 hours at 40,000 feet or 10,000 feet above Mount Everest, where the temperature is minus 40, minus 50 degrees. And he was only in a t-shirt and he survived. So that's freakishly hard to argue with, given that they had footage of him jumping into the wheel well in San Jose, and they see him then in Hawaii. So it clearly happened, and he clearly was exposed to an incredibly cold environment, and one that was also devoid essentially of the amount of oxygen required to sustain life. But yet he's alive. How did he do this? And you can hunt around on your smartphones and try to see if anybody has explained how he did that. And how, in fact, since the jet age began, 25% of people or so, if you go to Wikipedia, will show you that 25% of the people attempting these wheel well flights transcontinentally do survive. How do they do this? So that's where I'm headed. And so let me say that was my original, the way I got interested in it. But this was fun and everything, but it was at first difficult to rationalize how I would ever get money from anyone to pursue this, because you cannot suspend people without being arrested. That is, you can't put them into these harsh conditions and see how they do, because you're probably going to kill some of them, since only 25% survive in the wheel wells. So you're going to get arrested. And if you try to study the people to whom that have survived, by the time you've read this story, it's already over. So it seems an impenetrable place to go to study this phenomenon of the means by which people seem to have the ability to suspend and reanimate. So I was pretty much unable to pursue the phenomenon until I started reading about what happens when you have a heart attack. So this might seem odd that I would mention heart attack when we're talking about flying in a wheel well of a jet. But if you give me a minute, I believe I can convince you that similar things are happening. So I'm going to spend a little time now just describing my view of what happens when you have a heart attack. And to do this, I'm going to first tell you what you're doing right now as you sit in your chair, wherever you are, listening to me talk. Because it's going to I have to do that to give you a backdrop as to what's happening when you have a heart attack. So you're inhaling oxygen in the air. Your blood is coursing through your lungs, and some of the oxygenated blood is headed back to the left side of your heart. After passing through the left atrium, it goes to the left ventricle. The left ventricle squeezes and forces the blood out of the aorta, out into the aorta. And the first place that the blood divides, to eventually perfuse all of the organs of your body, is these particular arteries called the carotid artery, the coronary arteries. The coronary arteries take oxygenated blood and drive it down through the tree of the coronary tree and actually provide oxygen to the muscle that is beating. Okay, so think about it this way. The oxygen comes into the cells of the heart muscle, the left ventricle, and then it diffuses into the mitochondria, where on cytochrome C oxidase, the oxygen is literally burned or converted into water. And during that conversion into water, you're able to drive what's called the electron transport chain and make ATP. ATP is the universal molecule for animation. If you have it, you animate. If you don't, you don't. That's how it works. And it's weird, ATP is, because you have to turn it over about every 6 or 8 seconds in your body to resynthesize it. And so you're requiring oxygen all the time to resynthesize your ATP, because it's constantly being used to allow the actin and myosin to do what's called the contraction of your muscle to enable the blood to be coming out of your left ventricle. So you have no way to store oxygen. So you're fully dependent on the continuous supply, whether you're sleeping or awake at all times in your life, in oxygenated blood coming in so you can make ATP, so you can do the movements, the contractions of your heart to let blood continue to pump. So that has to happen. And you're doing it now. You must do it. And in many, many textbooks you've read about, if you stop doing that, you're dead. That's the thing I want to tell you about the baseline activity of your body. Now I'm going to throw a wrench in it and tell you that for short periods of time, several hours, if you stop having the ability to make ATP, you're not dead, oftentimes not dead at all. So let's now go through the natural history of a heart attack that you might experience. So the blood's coming into your coronary artery, but then suddenly a blood clot is in the ventricle, the left ventricle. It comes out into the aorta, and it goes into your coronary tree, into the coronary artery. And this blood clot or plaque blocks the ability for the red cells, the blood, to pump into the coronary tree. So within about 10 seconds or so, you don't have any oxygen in the cells of the heart muscle to enable you to resynthesize your ATP. And now a very interesting thing occurs. The ATP-ADP ratio, the amount of ATP in your cells drops. And now your heart muscle that was perfused previously by the coronary blood stops moving and enters into what I want to tell you is a state of suspended animation. This is not good for you because, if this is your atrium and this is your ventricle, what's happening is you suddenly have the ventricle wall not moving, and this wall might move and the atrium move, but this is now in suspended animation. And it makes the ability for the heart muscle to pump blood enormously bad. It's not very good at all because your heart muscle has now entered into suspended animation, but you're not. You're an animated person that has a great deal of needs, particularly for governor function in your brain, for example, to maintain life. So what happens as the blockage sits in your coronary artery and your heart muscle in your left ventricle enters into suspended animation, you will generally feel severe pain or fall to the ground. Hopefully somebody's near you and they get on their smartphone and they call for an ambulance. And what happens then is a bit of a race against time. What we're going to try to do is get you transported to a trauma center. And what they'll do is they'll put ECG leads on you when they are taking you to the hospital, and by telemetry they'll bring that information to the ED, the emergency department, and then they're going to say, "Oh my gosh, this person actually is having a heart attack," which turns out to be, in a technical term, ST elevation myocardial infarction, and they see that right on the ECG, and so instead of taking you when you arrive to the emergency department, you go right to the cath lab. And then what they do as quick as they can, they drive a catheter in through your groin, in through your femoral artery, and they thread this catheter up into your coronary artery. It's a puzzle to do it, but just imagine they can. They do it quickly. And then when they get to that point where they see the blockage using contrast, they actually inflate a balloon. And insofar as they do, they try to dislodge the blockage. And then they deflate the balloon, allowing blood to once again course through your coronary artery and reanimate. Now you're entering into reanimation of what was previously a suspended left ventricle. That's what happens mostly. We'll have some other details to fill in a bit. But I hope what you get now is that when you have a heart attack, particularly a severe heart attack, you are going to have your heart muscle enter into suspended animation where the rest of your body tries to remain animated. And it's a bit of a race against time to reanimate your heart. Now, by itself, you'd say, well, once you reanimate your heart, everything's good. Not so fast. Unfortunately, during the process of your body existing and your heart muscle existing in suspended animation, the reanimation, depending on the length of time, and it's usually on the order of hours between the block, this entry into suspended animation, the exit, balloon angioplasty. And during that time, your heart muscle that is in suspended animation adapts such that the means by which we treat heart attacks by balloon angioplasty results in excessive oxygen consumption in the early time after reanimation. And that causes the production of reactive oxygen species that chemically modify certain proteins and fats in your body, in the heart muscle itself. And that sets off an innate immune reaction that brings in your innate immune cells, such as neutrophils and so on, into your heart muscle. And as you're sitting there, having been out of now the cath lab, now your heart muscle is being killed off by your own immune system and results in you having what's called an infarction, or permanent loss of heart muscle forever. That loss of heart muscle, that infarction, oftentimes leads to remodeling of the heart, what's called wall thinning of your ventricle, and expansion of the left ventricle, which makes the pump not work very well. And then they start saying you have chronic heart failure, and then you die. And it takes about a year or two for a lot of people to die. Sometimes you can go on, depending on how big the heart attack, the infarct is. But that's sort of this state of affairs. So now you see that if I wanted to justify my existence to study miraculous survival stories, they're happening every day at the level of parts of people entering into suspended animation and in desperate need of a better understanding of the means by which we can reanimate more effectively to avoid the reperfusion associated permanent heart damage that's an enormous unmet medical need in the United States and around the world. So having done this very long-winded introduction, I'm now going to tell you about the study of suspended animation with a goal in mind, and that is to change the standard of care for heart attack medicine, to improve the ability to reanimate suspended heart tissue. So now, as I said, I cannot suspend people. It's just going to get me arrested. So what I could do is I could learn to suspend little things in the lab, like we have at the Carnegie when I work there, and they still study these things. And many labs in basic sciences around the world study baker's yeast and free living soil nematodes and fish embryos and such. and so I had them around. And so I began to study what happens when I take away the oxygen, now not from heart muscle, but from creatures that I just mentioned. One of the things we found out really quickly, that it's incredibly good. It's incredibly easy to suspend these creatures. You can stop the heart of a fish embryo really quickly by just dropping the oxygen concentration to below what's called the km of oxygen binding cytochrome C oxidase. It's about 1,000 times less than the amount of oxygen in room air will do it. You can also stop nematodes from doing anything that you can see under light microscopy at high resolution, and leave them there for a day or two, and they'll be fine. Then you just turn on the oxygen again and they'll reanimate just like you would turn the lights on in a room. Same thing with yeast. You can stop the cell cycle and so on right on a dime, and then restart it whenever you choose to do it. And that will last in a suspended state with not much loss of viability, even for a week. So this was really cool, right? I mean, I had a justification for studying an unmet medical need, and I had a basic sciences capacity to, if you will, conduct suspended animation experiments in these model organisms. And the way that I was doing it, of course, was this taking away oxygen. I thought, well, that's cool, that's neat. And, I got to thinking about it, and it turns out that while you're inhaling room air levels of oxygen, if you read enough about oxygen, you'll see that there's a bit of a dance with the devil thing. You want to inhale room air levels of oxygen. I would not recommend people go to oxygen bars. But if you do inhale an atmosphere that contains 5 times more than room air levels of oxygen, straight up oxygen, it'll kill you pretty quick. Oxygen is actually very toxic when you have too much of it around. So I thought about that, and I thought, I wonder if there are molecules in us or around that might restrict our ability to use the oxygen that we're inhaling. And just for your own edification, while you inhale however much oxygen you do, you only use about 20% of it as you run around your arteries and veins once. So most of it you don't use. So I wondered if there's anything that we may have in us that impedes our capacity to use oxygen, and if you will, buffer against oxygen so you don't burn too much or too little, and you don't have these reperfusion problems that happen because you're burning too much when you try to reanimate after balloon angioplasty. So what I'm going to tell you about now is agents that are in us, that are essential for us, that do that thing. The first one we found is a reduced form of sulfur called sulfide and a gas form called hydrogen sulfide. It's a heinous toxin. It was used as a chemical warfare agent, and it's required for life in you and I, and we make it all the time. So it's pretty weird, right? Why should we be making a chemical warfare agent? Well, maybe because of what I'm saying, maybe sulfide might buffer us against burning inappropriately, using oxygen when we shouldn't in particular cells at or a particular time. So when I found this molecule, I found it because I learned that people who inadvertently inhale too much hydrogen sulfide, because it's around in the air, you can produce atmospheres of this pretty easily. They actually have the experience of entering into suspended animation, but now not just their heart muscle, but their whole body. They just collapse. This phenomenon is called a knockdown. And the people deanimate, and they do have the appearance of being dead, but all you have to do is bring them out away from the sulfide, the hydrogen sulfide containing atmosphere, and they will reanimate without any problems. I thought, well, this is totally cool. You can actually control the state of animation in people. Now, this is really heinous to do this, and I'm not about to do it in people, but maybe I could do this in mammals, mice and rats. Maybe I could just, see if I can deanimate them and reanimate them, do knockdowns. So I got a can of hydrogen sulfide. I did this right after 9/11, and I did have Homeland Security actually did come to my lab. They've been to my lab because, as Joe said, I sometimes allow my enthusiasm to get the best of me. And so I started to get all these toxic gases, and therefore had to make sure I was screwed in right, not going to hurt anybody, which, knock on wood, I haven't yet. It's just really an important thing. But hydrogen sulfide, I showed a while ago, can convert mammals into reptiles. So I can take a mouse running around in a cage. You can see the YouTube video if you care. It runs around normally, but if I give it a little bit of hydrogen sulfide, not very much at all, then it will stop moving, having the appearance of being dead, like the guy in the wheel well of a jet, like your heart muscle in a heart attack. And it'll have the appearance of being dead, and I can leave it there for 5-6 hours, then just take away the hydrogen sulfide, and it'll reanimate and fire its jets, and it become a warm-blooded mammal again. During the time it's breathing the hydrogen sulfide, I can lower its core body temperature to like 10 degrees C, way below yours and mine, which is for warm-blooded mammals around 37 degrees C. So this is an animal that should have been dead, but it's not. It's fine. I should say that I found another one of these type of molecules that's also essential for us. We have to have it. And it's about 40 times more toxic than hydrogen sulfide. It's hydrogen selenide, H2SC. If you look on a periodic table, it's just, so it's oxygen, sulfur, then selenium. So it's one more step down. And this element, selenium, is essential to our lives, just like sulfur is. And it does exist in our bodies sometimes as sulfide, or in this case, selenide. And it too, I could use by presenting atmospheres to rodents, and I could convert them to reptiles and lower their core body temperature to less than 10 degrees and bring them back whenever I choose. Just flip on the lights, up to 6 hours later. So this was really striking to me. It meant that I might be able to control the state of animation in a free-ranging mammal. And so I thought, why don't I try to do this to control the state of animation of the heart muscle during a heart attack? That would be great, right? I could affect this reperfusion injury and help people out. Started a business. I carry pharmaceuticals, got a lot of people together. And you have to keep two things in mind with regards to using drugs. One is they have to be safe to use, and they have to be efficacious. They have to provide benefit to the patient. That's how it works. That's what the FDA looks for. In the case of sulfide and selenide, there was no question that they were efficacious. They worked like a shot in animal models, no question. but the amount of the drug that you had to use to create the benefit was very near to the amount that would kill the animals. What that's referred to in technical terms is called the therapeutic index. So the therapeutic index of sulfide and selenide were very, very poor. And the ability to conduct human evidence-based trials to show the usefulness of these agents to affect an improvement in heart attack was therefore lost, because no physician would give their patient these agents. Because when you did it, the amount you would give would be very near to that which would kill. And they would not do that because most people, over 95% of the people who have a heart attack, actually survive the heart attack event. We couldn't have people dying because of the therapy, right? That's just not the way it works. So that was really disappointing. That was many years ago. But I had this plan that maybe there are even more molecules than those that I had found, the sulfide and the selenide. Maybe I could find more things that might do this same thing and control animation. And one of the things that Joe didn't recognize about what I do, because I do jump around a lot, people wonder how I get from here to there. Really how I do is I just read a lot. Most of the time I just read. I don't really work much. I just read. And I was really shocked a few years ago when I came across a thing called the Bromide Sleep. No one really knows about the Bromide Sleep now. But you do know that when people are giving a poor lecture, someone, especially older people, might know, afterwards people say, God, that was really a bromide. And they're actually referring to something that's over 150 years old, when elements such as bromine were first discovered. And when these elements like bromine, and later we'll talk about iodine, were first defined as elements on the periodic table, people just ate some of them and saw what would happen. It was kind of goofy, really, but they did. And bromide, this reduced form of bromine, when you eat it, if you eat about as much of the bromide as you should chloride, and you know that's sodium chloride, that's salt, regular table salt. If you eat a few grams of bromide a day, you will enter into what's called the bromide sleep, and you will not be rousable. You will be relatively deanimated for upwards of a week or more. And published papers showing this are very clear. You can ask me and I'll send them to you if you can't find them yourself on PubMed. I was shocked to see this because it suggested that not just, if you're looking at a periodic table, not just the chalcogens, sulfur and selenium, had this control over metabolic activity, but also the halogens, below chlorine, that is bromine and iodine, again in the reduced state, that is bromide and iodide, would do this same thing. So now we're having 4 compounds, 4 elements, sulfide, selenide, bromide, and iodide, that for me, now I start to call them elemental reducing agents, or agents that are present in us that seem to be able to affect the ability for us to use oxygen. The really cool thing about bromide and iodide is they are phenomenally safe to administer both orally or intravenously. And this was totally cool to me because I realized, wow, the thing that I was tripping over was sulfide and selenide. They had the ability to control animation, but they were too toxic to use. So now we're going to go and see how far we can get using the halides, bromide and iodide, to actually get to the place we started, which is to make an inroad toward improving the reperfusion injury associated with heart attack. So this is a story that goes on for many years for me. There's a bit of a hitch that I want to mention, which is really interesting in terms of the natural history of these same elements that I just mentioned, before I talk about reducing to practice the ability for us to use an ERA or one of these elements to improve outcome in heart attack medicine. And it's that I became very interested as a biologist if these elements that I just named for you play a role in controlling the state of animation of creatures, then maybe I can look at natural systems that undergo changes in animation. And maybe I'll see changes in the level of these elements, these ions that I'm mentioning to you. And if that were true, it would really support the idea that not only have I found a way to do something in medicine, but I might have learned something about the natural means by which there is regulation of oxygen consumption in life as you are sitting in your chair right now. So for this work, I went to sort of extreme examples of alteration and animation that are present in the natural world. And I have for many years had a friend and colleague, Kelly Drew, who works on Arctic ground squirrels that she catches in the Brooks Range of northern Alaska every fall and studies them throughout the year as they go through their animated and deanimated states naturally as a result of maybe their association with incredibly cold weather that's up there in northern Alaska. So she was kind enough to send me blood samples from animals in the summer that are hopping around, and then the animals when they're in the winter hibernating with a core body temperature around 1 degree C and a metabolic rate that's about 100 times less than they are in the summer. And I was very pleased to see that one of the ERAs, iodide, showed high abundance in the blood. This is published. You can go read it on PubMed if you care. In the blood of animals that were hibernating, but compared to those animals that were running around in the summer and not deanimated. So this was really cool for me. I wondered also, since there's lots of evidence that people who have critical care injuries and diseases that bring them to the emergency department, but not heart attack, there's evidence that they don't have as much animation, sometimes loss of consciousness, such as a traumatic brain injury, or they might be septic. They may have been hit by a bus, gunshot wounds, blunt force trauma, that kind of thing. So given this work that we did in hibernating animals, we decided to see whether or not any of these same changes in the levels of these ions in the blood of ground squirrels were also present in the blood of people who were suffering what I refer to maybe as a near-death experience or very much a critical care situation. And this is the work that I've done with Dan and Ron and Jeff, who study in their respective places. Dan studies sepsis patients. He's an infectious disease guy at Harborview Medical Center, level 1 trauma center here in Seattle. Ron is the head of the American College of, past head of the American College of Surgery, and is the surgeon who is responsible for most of the surgery that goes on to people experiencing blunt force trauma. And as well, then Jeff Manley, he studies people with traumatic brain injury. And in all three cases, we are able to show, I think 2 of the ones on sepsis and trauma are published already, the other is not, increases in these same ions present in the blood of people experiencing these, if you will, experience-associated deanimation, that is sepsis and blunt force trauma and traumatic brain injury. So that is an aside, but it's important because it suggests that the agents I'm talking about are not just something I found that might do some good, but maybe we're already using naturally in our bodies to do good when we do survive lesser circumstances. So what I'm going to really talk to you about is now not just using these agents, but augmenting naturally existing responses to stress that human beings have to improve the ability to survive reperfusion injury associated with heart attack. I'm not going to spend a lot of time on it. This is not meant to be a deep science kind of thing. But over several years, well, for various reasons that I won't get into, I've chosen one ERA to work on, and it is iodide. It is the largest element on the periodic table that's essential to life for humans, that is iodine is. And I'm using it, and I'm giving it intravenously to, for instance, mice and rats and pigs that we put through a model of heart attack, where we deanimate the left ventricle and reanimate it, and then we ask how much heart damage or infarct occurs and how well the heart functions following this. And we do it without administering iodide intravenously to the animals at the point of balloon angioplasty, right, the clinically relevant moment, or we give the iodide. When we give the iodide, we're giving it in such a way that increases the concentration to even above the level that we see in normal stress responses in people having sepsis or blunt force trauma, because we've done dose response curves, and the maximum benefit is achieved somewhere between 100 and 1,000 times more iodide in the blood than is normally present in healthy people. When we do this, we can unequivocally improve heart structure and function. And I'm not going to talk about it any further. Ask me questions if you care. I'll send you the PDFs. So this then set off an effort at Faraday Pharmaceuticals to reduce to practice the capacity to change and improve reperfusion injury in heart attack patients. And I'm not going to spend a lot of time here. What you first have to do is if you mean to do this for real with evidence-based medicine, you have to have your preclinical pharmacology, which I just told you we have in mice and rats and pigs. Then you have to go on and show that your drug is safe. And for that, you have to do toxicology studies in 2 different kinds of mammals. We chose dogs and rats. And you have to show that you can give an amount of your drug that does harm those animals. And so you have to know what your ceiling is. And we did. And it is 30 times more than the amount we wish to give to help people who have heart attacks. So I didn't tell you this, but the dose that we have to give to a mouse or rat or a pig is identical in a mg per kg dose ratio. It's 1 mg per kg. That's really wild that it's scale invariant. So it was part of the reason we chose iodide. And what I'm telling you is you can give animals for 14 days in a row an IV bolus of 30 mg per kg in these 2 animal species, and you do not create toxicity. So that means the therapeutic index or the safety range for giving iodide IV bolus, if you make a mistake and give it to them twice, doesn't really matter because you got to give them 30 times more before you begin to even think about seeing toxicity in these 2 different animal species. So the FDA says, okay, you've convinced us. We'll let you administer this to people when they actually have when they're just healthy volunteers. This is called a phase 1 safety study, which we did a few years ago. 40 people got various doses of iodide, and we showed we could give the therapeutic dose without having any problems in the people. Then the FDA said, now you can do a phase 2 trial where you give a dose that you intend to help the people, but they have a disease, but you cannot do it as a closed perspective. You can't do it as a blinded study. It has to be open label, because if there's a problem, we need to know about it right away, and we're going to shut down your trial. So at 20 some odd sites in Europe and the United States a few years ago, we actually gave 123 people the dose of iodide that I just mentioned. And we did see that it was safe to give. And we did see that there was benefit in those people based on heart structure and function using a 3-month MRI, which was acceptable as an endpoint to the FDA for a glimmer of efficacy. Before you run around and say, my God, he's cured heart attacks or something, or reperfusion injury, let me mention that the numbers of people you have to study to get past what's called type 1 and type 2 errors to be sure that you have efficacy, we did not have in our study. It was just a safety study. And while we saw strong trends to decrease heart damage and improve heart function, we cannot say that we do not have a type 1 or type 2 error. I don't want to get into what that means, but just say we have not really proven it yet. But the investors in the business that I founded a few years ago is sufficiently intrigued that there's a benefit to be had, that we will begin a phase 3 pivotal trial at 150 sites in Europe and the United States that does have 2,500 patients enrolled in the next 2.5 years. And that will enable us to either see or not whether there is truly a benefit that is getting past type 1 and type 2 errors, and the fact that human beings have blockages at different parts of their tree. They have different comorbidities, they're different age. All this contributes to variation. But this variation is understood, and we know that the numbers of patients we're enrolling will tell us yes or no whether this has been a change to practice to improve outcome for people suffering reperfusion injury and heart attacks. So I'm kind of done talking now, but I will just say a couple of things in closing. And that is just to say, I've been very interested in these near-death experiences and the survival, these miraculous survival stories you read. And if someone were to ask me, what was that 15-year-old doing in the wheel well of a jet? I wonder whether it wasn't what he ate Because by the way, if we give some animals more of one of these ERAs just in the diet, just have it, eat it or drink it, then we can show that they do better. And so it could be that you're more dependent on what you ate recently than you might realize. Now, having said that, please do not run off and eat a bunch of iodide. There are drawbacks. I would feel very bad if that's what you got from this. So please do not do that. But eat a healthy diet, and possibly this 15-year-old who managed to fly above Mount Everest at minus 50 degrees in a t-shirt and survive after 5 hours happened to have the ability to redistribute ERAs in his blood that allowed him to, if you will, hibernate or suspend appropriately in a way that he could naturally reanimate. And while I don't know that that's true, if somebody asked me what might have happened, that's what I would say. And now I think I'm done. So thank you for listening. I hope you were entertained, and I'm happy to answer questions, which I said I would do during the talk, but that was actually virtually impossible. Okay, for those in suspended animation, how much ATP must be produced to continue health? Another word is induced coma in the context of astronauts for lengthy time travel. Oh, this is someone asking about time travel. Cool, that's a great question. Let me say something that I neglected to say in my talk, and that is that when the heart muscle goes into suspended animation, when the animals we study go into suspended animation, they actually can't do oxidative phosphorylation to make ATP, but there's kind of a backup pilot-like means by which you can do it. It's called glycolysis, and it involves a reliance on glycogen that's stored inside of your cells, and you don't really make CO2 and water out of the burning of the glycogen because you don't have any oxygen, but you can get little bits of ATP. And that's what we showed sustains the animals and likely your heart muscle when you're deanimated and not doing oxphos. You're reliant on this internal store of glycogen. So I think we've tried to work some on space travel in the sense that, you know, if you combine hypothermia, that is cooling, really severe cooling, with the no oxphos, no oxygen, or not enough oxygen to do oxphos, it's quite a positive thing. And I do think that there may eventually be people, well, probably won't be me, who can take advantages of this for what you call other things besides medicine, whether it's diving or going to high altitude, or in fact, going all the way out to the atmosphere. So we'll see how that goes. Anyway, the next question, it says, for our astronauts living in space station for 6 months, come back to planet Earth and find alterations in their vision going into space. Their vision is 20/20. Upon return, it's 2400. Yeah, so this is a person, I think, asking me whether or not re-entry into a gravity-containing atmosphere like the Earth from space station, might you benefit from understanding something about the metabolic flexibility that we're understanding from suspended animation? It could be, but of course they're not breathing, you know, they're breathing room air levels of oxygen up there. So it's not directly related, but I can understand what you're getting at. Maybe if you could think about just the metabolic flexibility that occurs when your environment has changed, maybe this is an entree into studying when your gravity has changed rather than oxygen levels. And that's interesting, although I have not pursued that. Oh, sorry, here's another question. This is David Conover. Seems every individual is a little different. Can you comment on how fitness as a variable, which impacts one's VO2 max, oops, something went away really quickly. VO2 max might have an impact on what you are suggesting regarding animation? Oh, so David, you're going to have to talk to me offline because you're getting into VO2 max and that's oxygen use and so on. I'm not going to be able to answer that one. I can, but I don't think it would really be good for this Q&A. So please, if Naveed can get you my email, I'll answer that one. That's a tougher question on the impact of fitness and VO2 max. Here's one from Nurse Jane Swan. For our submariners who just sustain themselves at depths greater than 800 feet below surface and who unfortunately sink to stable surface, how do we preserve life until rescue? Oh, if somebody gets trapped in a submarine, like it's happening, maybe, well, it happens occasionally. Yes, I think we might be able to make a difference for people who are in situations such as trapped in a submarine. But of course, those sorts of circumstances are so rare that, I don't know how to quite get to that place. I am studying people who go to high altitude very quickly and get acute mountain sickness, both with the US military and also the Altitude Research Center in Denver. So I'm doing some of that. And I also study hypoxic people who are made hypoxic at the UCSF with a guy named Phil Bickler, who's an anesthesiologist there. So we're trying to get into it, but I don't think we're going to just, and it could translate to help for people who are trapped in submarines, but not immediately. There's a lot of work to do between here and there. Here's one from an anonymous attendee. Is there any way to know if the iodine is a side effect or direct activator of the reanimation? Oh, I can tell you that one. So iodine or iodide is not an activator of reanimation. That is true. If you put it into an animal that's deanimated for low oxygen, it doesn't reanimate. What does happen, though, it's very curious. Iodide is able to degrade the reactive oxygen species that happen when you overplus during reanimation. That is, it is able to function, like for those of you who are scientists, it functions as the enzyme catalase and catalytically destroys hydrogen peroxide. And you can see sort of YouTube videos about that. So I don't think it's a direct activator of reanimation, but I do think it's blunting the ability for these reactive molecules during reanimation to kill off the heart muscle, to send in the goons, as it were, the innate immune reactions that kill off your heart muscle. Here's one from Niels Nielsen. Have you or anyone studied extreme free diving? Oh, this is great, to see if it also involves similar reanimation mechanism. Oh yeah, we're starting to do this on the Weddell seals and the pearl divers. So I'm very anxious to see if they're having redistribution of ERAs and that sort of thing. Now, I don't know the answer to that right now, so I can't offer you anything, but we're very encouraged by collaborations we currently have with people studying the Weddell seals and pearl divers. Here's one from Nurse Jane Swan again. Please, if you can, do you work with scientists in the Russian Federation? I don't understand that. That's not really a question. I don't work with anybody from the Russian Federation. But anyway, if you want to get an email from Naveed, maybe we can talk offline. I'm not sure what you're really asking there. Here's one from Don Powers. Any clinical research studies with IV administration of potassium iodide, titrated, of course, finger pulse oximetry monitoring, nasal oxygen, also clot busting enzymes administered either IV ASAP or in the cardiac cath lab? Oh, well, Don, this is great. First of all, of course, we never infuse potassium iodide because potassium is very bad. You have to use sodium iodide because you wouldn't want to defibrillate the heart. Anyway, it's very bad for heart muscle to get potassium very high. We have done some of the work you're mentioning, and it's a bit too technical for this. But again, if you get email from Naveed, I'm happy to talk to you in detail about that. It's a lot of work in there. Yeah. Donna Rogal, MD. Might any of this be related to chronic fatigue syndrome? Well, so it's really interesting you mentioned that. There's a lot of alterations in animation that occur in people that aren't so acute. And you're mentioning one, chronic fatigue syndrome. And it could be, and I think what's going to happen is that more people will get wind of what I've just talked to you about, and they'll do this. They'll see if there's correlations between, changes in the levels of ERAs and people who have CFS. They may, and then we could maybe think about how to adjust them and affect maybe their physiology, because maybe they're in a permanently kind of deanimated state, walking around kind of foggy and incapable of having the degree of animation that most people have. And they'd like to be more animated. So they'd like to have diminishing the levels of these impacts of these ERAs. So I think that's a really interesting thing, and it could happen. Dean Callahan asked, we can eat a bunch of seaweed, which is high in iodide. Oh yeah, so Dean is mentioning that the element iodine is not just present in your Morton salt that you have at home. It's actually present. It was first discovered as an element by virtue of the fact that it's very high concentrations, iodine is, in certain types of seaweed. And you can certainly eat it. I want to return to that thing I mentioned in my talk. Please do not run around and eat a bunch of iodine because you heard my talk to fend off a heart attack you may have in the distant future or whatever. Please do not do that because there are negative side effects with eating huge amounts of iodine for long periods of time, and we can discuss that. But if you're willing to take my word for it and just please do not eat a bunch of iodine. Please speak to your doctor before you start eating a pile of iodine. It would really be bad. You shouldn't do it, and I hope that gets across. An anonymous attendee says, "What is the source of the free iodine in mammals? How is it mobilized to respond?" Yeah, this is a great question. How do you mobilize it in traumatic injury? Well, we don't know much. Iodine, when you eat it as iodide, well, it goes into the blood like chloride does. It's concentrated in different parts of the body. The main one is your thyroid gland in your neck. It is used there to form what's interesting, a kind of a hormone called thyroid hormone, thyroxine T4, for those of you who know. And it comes out of the thyroid gland, and it's actually a go hormone. It's a thing that stimulates you to burn and to do things. So it's curious that iodine, 4 atoms of iodine, or T4 for the 4 atoms of iodine on essentially dityrosine, is thyroxine. And then iodine atoms are removed to make T3, which has the most activity for going. And then there are other forms too where enzymes remove iodine atoms. And of course, when they do, they create free iodide. So it could be reasonable to think about the fact that the increase in iodide in the blood in the trauma patients is coming because people are, their enzymes in the neutrophils and other parts, they're degrading the thyroid hormone, and thereby creating a product called iodide, which has this other chemical activity. So it could be that instead of just having thyroid hormone be a goer, now this is a going down thing. So for example, if you look at neutrophils and you give them a reason to have what's called, to get mad and to start to become active, what's called oxidative burst, you'll find that thyroid hormone stimulates oxidative burst. But actually iodide suppresses oxidative burst, okay? If you look at neutrophils taken from people, before they get a hit of iodide, they have a certain ability to get mad and do oxidative burst. But it's published that if you give them iodide for 3 days at the sort of the dose that I'm using, and then take the neutrophils out again, the neutrophils are very quiet, okay? So this points something out, right? You now have a control over the innate immune reactions based on how much iodide you eat. But be careful with that, because you need your innate immune reactions so as not to get some sort of, opportunistic infection. So it's not a panacea. You're messing with something pretty powerful when you eat iodide. And it may be connected to this thyroid hormone and deiodination, which is another subject that I don't have time to go into. What is this? Anonymous attendee says, what is the source of free iodine in mammals? Okay, sorry, we just answered that. Okay, Ryan Gleason asks, how does this relate to deep hypothermic circulatory arrest used during it? So sort of when you have killing people on the table for clipping an aneurysm or something. Yeah, so there is this, I don't know how it relates, which of course there are people where you do full circulatory arrest to clip a brain aneurysm or whatever. And I don't know how it's related, but it'd be interesting to figure that out. I just don't know. I know people who do it, and I could get blood samples and see, but I haven't done it yet. Don Powers asks, excellent knowledge concerning adjunct chemistry necessary in the body to assist in lower heart muscle cell death. I'm sorry, Don, we're gonna have to talk about this later. I'm not sure what you're asking, sorry. Nicholas Zill, what is the role of low temperature in modifying reperfusion injury? Well, it's a complex issue. Low temperature does improve outcome following cardiac arrest in people. That is where your heart stops and they turn it back on. It's been shown 15, almost 20 years ago. It's a standard of care. It's pretty heinous. You have to lower the core body temperature of the people. A lot of people don't want it done to their loved ones. It's not been adopted very well in the community, but it does if you treat 7 people with hypothermia following cardiac arrest, you will save someone's life. And that's been shown by evidence-based medicine. So there's something powerful there about hypothermia, as well as difficult births for babies. They've shown the same thing, a Detroit study many years ago. But the understanding of the connection between low oxygen and low temperature is the frontier. And I like that. I think a lot about that. But whether it's space travel or doing better in these cardiac standstill surgeries where hypothermia is employed, because there are a lot of hypothermia trials that just, in people that just don't work. Hypothermia just doesn't work. But in these 2, cardiac arrest and difficult births, it has worked. And that's about all I can say. It's just something we should all work on, but I don't have any time. Lee Buchanan asks, during the period of suspended animation, is the process of aging also retarded? I love this question. This is great. So in a sort of Einstein kind of speed of light kind of twin paradox kind of way, if you can have that in your head, and I don't have time to really go through it. But, one of the twins is aging, the other isn't. You get the idea. Sort of a bit of a Rip Van Winkle kind of thing happens. Well, if you do suspend nematodes on a Monday at the 2-cell stage, and another twin from the 2-cell stage has been also taken up, but it isn't suspended, then you can come back a couple of days later, and the twin that was animated is already having babies that are older than its twin that was suspended. And then you can reanimate the twin that was suspended, and it'll go on, but it's 2 days behind its sibling. And given that you deanimated on a Monday and you reanimated it on a Wednesday, it becomes a really interesting question whether the suspended animal had a Tuesday. And that gets into eigentime and self-time and time travel to the future, and whether or not suspended animation will enable us to travel in time to the future or not. And that's about as far as I can go, but we can talk offline if you care.
Wanted to cut in and give you a 5-minute warning here, so we have a time for a few more questions.
Okay, well, I'll go and then I'll stop.
Okay.
Jimmy Best asks, are there supplements that may be useful for benefit the state of animation? At this point, no, there aren't. As I mentioned, don't start eating a lot of iodide. So not at this time. Anonymous attendee, have you had success with suspending organs for training? Yes, we have. Some of this work is published. Unfortunately, I don't have time to go on about it, but done this with flight surgeons, the U.S. military. It's published where I've been working with guys at Harvard. I'm sorry, I don't have time to go on about it. I'm just going to race through some of these quick. Paul Rodriguez, can ATP itself be injected? No, it can't, because it doesn't cross cell membranes. So it doesn't work. Sorry, Paul, that one doesn't go. If approved, what is the emergency response procedure for preventing heart damage after trauma applied by medics, doctors? Oh, I think we might get into crash carts. I think that what might happen, because we're doing a trauma trial, phase 2 trauma trial for blunt force trauma, getting hit by a bus, that kind of thing, in a bunch of sites across Europe and the US by infusing iodide. And we're doing it right in the ED, in the emergency department, and every day thereafter as they exist in the ICU. But I think what will happen is it's so safe. The first responders are just going to give this prophylactically. When someone's bad enough to be laying on the ground and, cardiac arrest, blunt force, gunshot, whatever, I think this could easily become a standard of care just right in the field right away. Does using iodide avoid iodine allergies? Well, there are no iodine allergies. And Boris, you can get it's a misnomer. You have to just get that from email from Naveed, and I can talk to you about that. Anonymous attendee, are you aware of the mental state of a person that enters suspended animation? Well, the short answer is no. Although, when you do talk to people that have been suspended, they tell you they have no memory. So, yeah, they don't have consciousness for sure, as you've asked. Peter Kessler, first, thanks so much for this life-saving research. Well, if it works, I'll agree with you. It might. Overall, as we nail this, what are your educated speculations for the length of time suspended animation of humans during extremely long distance space travel? Well, I think if we combine with hypothermia, we could do well. Hey, look at this. This is somebody I know. Margaret, hi. From up there in the UK, there in Scotland. Have you looked at cultures where people eat lots of seaweed? Yes. And see if outcomes and heart attacks are better. Well, I will say the people who eat the most iodide live the longest. And hi, Bill and Margaret. How are you? Fraser kindness. I'm sorry, not pronouncing your name right. Curious about the circulating ERAs in patients with hypothyroidism. Do these conditions alter circulating iodide changes? We haven't tested that. For people like post-injury, we don't know. Anonymous attendee, would it be interesting to see if this process is at play in deep dive? Yes, it would be for deep diving mammals and porpoise. We haven't done it. Scott Mitchell, would any of the compounds you have discussed be trigger for the hallucinations that some people experience with near-death experiences? Possibly, but I don't know. Anonymous attendee, would people with hypothyroidism be more susceptible to having adverse outcomes and heart attacks? Possibly, but I don't know the answer. And lastly, an anonymous attendee asks, would people without thyroids be expected to react differently? Yes, but I've not studied it. Anyway, thanks a lot, guys. I am tired, and I appreciate your listening.
Thank you so much. That was great. And if we didn't get to your question, or if you want to ask more questions, I put our email in the chat, and at emb-communications@carnegiescience.edu. Email us your questions and we'll get back to you soon. Thank you all for joining us.
Take care. Bye.
Thank you, Mark, for such a wonderful talk.
Take care. Bye.
Bye-bye.